Finally, the standard membrane-based model of the neuron, based on ion channels, fluxes, mediating voltage gradients, is dubious. And I'll talk a little bit more about this. This is based on some very interesting work by Gerald Pollack. But first, let me talk about number 4. Here's a paramecium. It's going to bounce into something, retreat, and go in another direction, all based on these cytoskeletal ciliary structures. And it can find a mate and merge with it and have a sex life, and it doesn't have any synapse. It's just a single cell. It can also learn. If you put it in a capillary tube and let it escape and put it back, it'll get out quicker each time you do that. It does it with its internal cytoskeletal structure, and neurons have a similar, even more complicated cytoskeletal structure that is kind of not just the skeletal support system, but some of us believe the nervous system within the cell also. We'll come back to that. And this is what the inside of a dendrite actually looks like. It's not a bag of water, but a highly ordered network of its own, kind of like a fractal subdimension, a network within a network, or a forest within a tree, as somebody called it. Okay, let's get to Pollack's work. And I was taught in medical school, and I'm sure everybody was taught pretty much about these ion channels and the membrane runs everything and so forth. And it's based on some rather dubious assumptions. If you look at the very, very top, A, this is a patch clamp where a pipette has a piece of membrane and measuring voltage across it. And you get these little blips of voltage. So these are thought to be ion conductances. Well, it turns out if you put a silicon rubber patch, you get the same thing. And if you put a polyethylene filter, you get very much the same thing. And if you put a pure lipid bilayer without any proteins, you get a very similar thing. So it's not ion channels opening and closing. Something else is going on. Also, if you take a cell, punch it full of holes with electricity or detergent, so that all of the potassium should leak out, all of the sodium should leak in, it should die pretty quickly, but it doesn't. It hangs around for quite a long time. And in fact, you can use detergent, take the membrane off completely, and it still does fairly well for a while because of the gel structure of the cytoplasm. Cytoplasm is not a bag of water with things floating around like a minestrone soup. This is actually a schematic of all of the structure inside a cell. That stripe would be the membrane, and then all the other structures are proteins which make up the structure inside of a cell. So there's not that much room for the water. There is water in the spaces. But because of all this surface, what happens is the water gets ordered. And because there's charges on all these protein surfaces, and the waters form something like, it's not really a crystal, it's not like ice, but it's not really aqueous bulk water either. It's somewhere in between, a pseudocrystal, if you will. Now, the reason this is important is that potassium, for example, is soluble in this ordered water, but sodium is not, because sodium is too big, which means that if the water is ordered, it's going to hold the potassium in, and it's going to exclude the sodium. So this could explain why we have this gradient across the cell boundary, irrespective of ion pumps. So if we have a transition in the protein, it's going to disorder the water, and the solubility of the sodium and potassium is going to change, and this can account for fluxes. And Pollack makes a beautiful argument for this, and I recommend his book highly. And by the way, he'll be at the conference that John mentioned in March here in Tucson. Similarly, vesicles, including neurotransmitter vesicles and so forth, can also be accounted for by the same type of mechanism. So what about receptors? A lot of psychiatry and anesthesia is based on drug effects on receptors, which presumably open ion channels. Well, his story is that if you look at the bottom, we have transmembrane proteins which undergo conformational change in the top and flip and carry ions with them. So the ions don't fall through a hole, they're actually flip with the protein as it flips back and forth across the membrane. Now, I actually wrote a review of Pollack's book for Amazon because I liked it so much, and I said something like, if this book doesn't get burned, it's going to be a classic, because it's so heretical that I think a lot of people are not going to like it. But I've sort of thought there was something fishy about this whole story, and this book gives a good argument that there may indeed be. Similarly, if something like acetylcholine binds to a receptor, there are ionotropic receptors that open ion channels, or supposedly open ion channels, and also metabotropic receptors that have direct connections inside the cell to the cytoskeleton, And Nancy Wolf and I, in a paper in Trends in Cognitive Science, described acetylcholine decoupling the microtubules from the membrane by these actin bridges, which would serve to promote a quantum state, which I'll get to later. The point is that metabotropic receptors are as important as ionotropic receptors, and by direct effects into the cell itself. If we just think of the cell as a gel system rather than as a membrane, as a bag of water surrounded by a membrane, and just look at the gel characteristics, we get some very interesting stimuli response characteristics. These are pure gels, not even biological gels, just gels. pH, temperature, chemical, biochemical agents, solvents and salts, electrical field, electromagnetic radiation, mechanical stress, elicit all these responses: chemical, phase separation, shape change, surface, permeability, mechanical, optical, and electrical changes. So the gel characteristics of cells may be extremely important, and they've been eclipsed by everybody focusing on the membrane, and for good reason. The membrane is certainly important. But I think in the future we're going to see a lot more emphasis on this. Well, that brings us to the question of drug action, and psychiatric drugs and so forth are extremely important, SSRIs and many, many others. This is from Feldman's Fundamentals of Neuropsychopharmacology, listing 7 sites for drug action, psychiatric drug action. And I'm going to skip ahead and give you the numbers. 1 are microtubules, which are inside the cell. I showed you those before. 2 would be the excitable membranes, including the proteins. 3 sites for synthesis, sites for active uptake, such as the SSRIs. And I've always been curious why antidepressants, the reuptake or uptake effect is immediate as far as I understand, but the clinical effect takes weeks to months, if not longer. And someone explained that what happens is the cytoskeleton, which after all regulates and controls the sensitivity of the synapse, gets restructured, and it takes that long for it to be reprogrammed and restructured. So it's the cytoskeleton in the cytoplasmic gel system which is controlling the synaptic strength and whatnot, which accounting for the delayed effect of SSRIs and other drugs. Amphetamines, on the other hand, and cocaine and whatnot are most likely immediate effects, because that effect indeed is mediated by prolonged catechols in the synapse. And agonist, antagonists, organelles, and so forth. So the point is that it's not all receptors, and we need anesthetics. I should mention. For many years, were thought to work. Most of this century, were thought to work in the lipid part of the membrane because their potency is directly correlated with solubility in a lipid environment. But in the 1980s, it was discovered that anesthetics work directly on proteins, ion channels, receptors, and other proteins, and that there are pockets within proteins that are lipid-like, called hydrophobic pockets. And it turns out that the anesthetics act by affecting quantum mechanical events in these little hydrophobic pockets, which are something like the brain of the protein, if you will. So we know that inhibits consciousness, the anesthetic gases. And the story is still open on a lot of other drugs, but it's not strictly agonist-antagonist at receptors. It's a bit more complicated than that, and probably at least secondary effects on the cytoplasm and cytoskeleton. The other question, is upon what level of brain organization does consciousness critically depend? Well, most of the emergence people would say neurons are the fundamental bits, but you might also say, well, brain regions, for example, prefrontal cortex has certainly an executive function. Neural network coherence, we know that the best correlation electrophysiologically is with a coherent 40 hertz gamma frequency. Particular neurotransmitter systems, acetylcholine seems important, at least in attention. Collective synaptic activity, ionotropic, metabotropic, and also electrotonic. There are many gap junctions in the brain, these electrotonic portals between cells, which, and it turns out that the 40 hertz oscillations are mediated by gap junction electrical conductivity, electrotonic activity, rather than any chemical, accounting for the almost perfect phase coherence across the brain of 40 hertz. Other people look at axonal membrane spike trains as the currency of consciousness, if you will. Also dendritic fields, the electrical fields around the dendrites, which mediate EEG. or intracellular processes, second messengers, cytoskeleton effects, and so forth, and quantum coherent processes, for example, those in the cytoskeleton. So all of these may have some effect or role. Well, this is a partial list of theories or neural correlates of consciousness modified from one of Dave Chalmers' papers. He calls it the zoo, like the particle zoo of consciousness theories. Some of them are just neural correlates or cognitive correlates. A few of them attempt to actually explain the hard problem and other problems. Perhaps the best known, or one of them, is Crick and Kolk's idea of 40 hertz oscillations in cerebral cortex, because it does seem to correlate best. And we have some anatomical things like activity in intralaminar nucleus in the thalamus, reentrant loops in thalamocortical system by Gerald Edelman, 40 hertz activity in thalamocortical systems, activity in nuclear reticularis, extended reticular thalamic activity, Newman and Bars, anterior cingulate system, neural assemblies bound by NMDA receptors, Ben Libet's idea of temporally extended neural activity, back projections to lower cortex, extrastriate, prefrontal projections. Crick and Kolk think that visual consciousness does not occur in V1, but actually is projected to prefrontal areas where it becomes conscious, and they have some evidence for that. Neural activity in area V5. Beck and Eccles, John Eccles, the Nobel laureate in neurophysiology, in his later years got into the problem of consciousness and adopted a quantum approach with Frederick Beck. Eccles was actually a dualist, and his idea was that the part outside science or God or whatever he was thinking acted at synapses by this quantum indeterminacy problem. Beck is not a dualist, but thinks that quantum indeterminacy happens at synapses. Walker has this old idea of quantum tunneling through synapses going back to actually to 1970. Logothetis and Schall actually did some extremely interesting, exquisite experiments with monocular rivalry in monkeys to basically what they did. Well, actually, I'm going to show you a little data from there in a second, so we'll come back to that. Susan Greenfield's neuronal gestalts, Jeffrey Gray's comparator system. In 1994, I published the idea that quantum coherence in microtubules was the substrate of consciousness. Bernie Barr's global workspace, thalamocortical global workspace, activated memories, and so forth. Roger Penrose, in 1989, wrote a book called The Emperor's New Mind, where he suggested that our conscious minds did things which were non-computable, non-algorithmic. That allowed us to be different from computers. And the only source of this non-computability was a type of quantum collapse of the quantum wave function based on quantum gravity. And I'll come back to this idea a little bit later, but it's certainly quite different from most of the others. Ian Marshall, in 1989, suggested a Bose-Einstein condensation of brain receptors, and Bose-Einstein condensates were actually proposed by Bose and Einstein in the 1930s and then realized in the lab in the 1980s or 90s. Basically a condensation of a coherent state where all the members become unified. And he proposed it as a solution to the binding problem. In 1995, Roger Penrose and I published the first of a series of articles suggesting that his quantum gravity objective reduction occurred in the microtubules in the brain. I'll come back to that later. Others have suggested electromagnetic fields in the brain are consciousness. I mentioned emergence complexity arguments. Al Scott, who used to be here at Arizona, and a number of others. Carl Pribram in 1972, The Brain is Hologram, which had some interesting appeal. And anatomically, Nancy Wolf and I published in Intrinsic Cognitive Science the idea that rather than this ascending system of consciousness was in this horizontal syncytium of dendrites interconnected by gap junctions oscillating at 40 hertz. I mentioned Logothetis' work, and they did some very interesting studies in monkeys. They trained these monkeys to look at metastable objects like Necker cubes, so that you look at this thing which could be, I forgot to make a slide of it, but I'm sure you've seen it, where the surface could be back or forward. And our perception, our conscious perception, goes back and forth between the two. And they trained the monkeys to somehow report which one they were seeing, and then they had electrodes to see, and they found this sulcus region, I forget already what it was. superior temporal sulcus activity showed the actual, whichever one they were reporting. So visual cortex had both, and then whichever one, and it flipped back and forth in this superior temporal sulcus. A very beautiful neural correlate of consciousness.